Hi everyone, welcome to my talk on predictive intelligence for network neuroscience methods and challenges. So what is predictive intelligence about? Let's look at these first questions. Wouldn't it be terrific if we can diagnose early and better with minimal resources and at lower cost, which means that we have a limited data and still we can diagnose with high accuracy? How about we collect less data and predict more? So instead of collecting more medical data, we can think about generating and predicting data that is biologically sound. The third question, or the third point that we try to address through predictive intelligence, is to generate medical data across different dimensions or domains, and I will explain this in the next slides. So let's say, for example, here, let's imagine if we have a T1 weighted MRI, an image, and then from the image, we derive brain connectivity. So this is what we introduced recently, the notion of a morphological brain network that is derived from structural MRI. So from this network, we can predict the brain connectome or the brain network evolution across different domains. So for example, from morphological connectivity, we can get functional connectivity, diffusion-based structural connectivity. And also we can try to generate brain networks across resolutions, which means we have the ability through the predictive intelligence to generate higher resolution brain connectivities. The third domain or dimension here is time. So given a single observations, we can predict the evolution of the brain across time. So at my research lab, the BASIRA lab, we focus on, primarily we focus on designing new methodologies rooted in deep learning and geometric deep learning for predicting the evolution of brain networks. This is across time, also predicting brain networks across domain and resolution. So we have these three dimensions. And today I'm going to just give you an overview of a few works from the lab. So the first one was presented in MECAI 2020. So how to generate brain multigraphs from a single observation. The second one was presented recently at ITME and selected as oral presentation. How can we super resolve brain connectivity networks? The third one is can we predict the brain evolution network across time from a single time point? And this was presented at Prime MECAI 2020. And there is a fourth dimension that permeates these three, which I call the brain graph template or the brain network template. And this will be presented, I will present the work by my student who got also his paper accepted in MECAI 2020 and extended into a full journal paper. So let's start from the beginning. What are these three predictive intelligence dimensions? So the first one, as I mentioned, is domain. So imagine if we have a single source brain graph. Okay, and then what we aim for, what we want is, given a single graph, we want to learn how to predict a target graph. So for example, for morphological connectivity, we can predict functional connectivity. So our goal is to learn this mapping function or this transformation F that maps one input domain into a target domain. And another problem, a more complex problem that we can solve, is to predict multiple domains or multiple graphs. So instead of predicting single modality, for example, more functional connectivity, we can also predict structural connectivity. So we're generating or predicting for the same subject, for the same brain, multiple connectivity maps of that brain using deep learning methods and advanced learning methods. So across resolution, the idea is given a low resolution graph where the brain is parcellated into coarse anatomical regions, we try to learn how to predict the parcellation of the network at a finer scale. So this is what we call super resolution. So here we have a larger number of nodes, and we can see how the connectivity between these nodes have changed. So the third dimension is time. So time is so important because if we can foresee the evolution of a single brain graph at a particular time point, we can detect abnormal or atypical changes in brain connectivity. And these will consider biomarkers for particular neurological disorders. So prediction over time is very important for predictive medicine. So here our goal is to learn a time-dependent function that allows us to predict the evolution of the brain connectome or the brain connectivity map over time. Now, the last or the fourth, task or dimension component that permeates these three dimensions is the concept of graph integration. So here what we have, imagine that we have different subjects. So each subject is represented by different brain views. These could be multimodal brain connectivity. So for a single subject, I have the green connectome, the blue, and the pink, and these three capture different types of brain connectivity maps, like function, cognition, structure, morphology, etc. And the goal here that to generate a holistic integral map of the brain by learning this mapping, and this is what we call a CBT or connectional brain template. Now, this connectional brain template can be used to predict, basically super-resolved brains, generate multimodal or cross-domain brain graphs, and also, predict the evolution of brains. So the template is so important because a template acts as a fingerprint. We can use it to guide our learning algorithms across these three dimensions. Now, eventually, the goal after we predict the data is that we use the predicted data along with the original source data to for disease fingerprint prediction, for example, for biomarker prediction, for disease classification. So we try to classify a disordered and normal control subjects. So there are lots of applications, meaningful clinical applications that we can do using predicted data. So all these works are available on the BASIRA Lab YouTube channel and also the BASIRA Lab GitHub. So all the codes are free and you can feel free to test them. And these do not only apply to brain connectivity, as long as you can model your data as a graph, then you can apply them to your data. So let's start with the first one. So the first one is like cross-domain. The first example is cross-domain prediction. So for cross-domain prediction, I'm going to give you the example of here. We have like a topology-aware generative adversarial learning for multi-graph prediction. And the code is also available on GitHub, and the full presentation, if you would like to learn more about these details, please have a look at the YouTube video. So what is the concept here? What is the problem that we would like to solve? So given a single brain graph that we get from an MRI data, for example, resting state functional MR, we aim to learn how to predict multiple types of brain connectivities, maybe derived from the same modality or other modalities, okay? So this multi-graph generation task is our goal. Now, if we look at existing works, they generally overlook the local topology of the graph because we know a brain network has a topology, it has hubs, it has modularity, etc. And mostly these methods, they are limited in scalability because they work on source to target prediction. They don't work on joint multi-target domain prediction. So what is the model or the solution about? So we create or we design a graph neural network architecture. The input here, we have a source domain S. So all these are our subjects, and we have the target domain, and these are the training samples. So the first thing we do is like we vectorize each connectivity matrix of the brain graph, and here we stack it. So we have the brain graphs represented for this source population and this matrix. And we also learn what we call a similarity, a subject to subject similarity matrix. And this matrix is important because it tells us which subjects are similar, are clustered together. And we know if they are similar, maybe in the source domain, they would also be similar in the target domain. So this is a strong assumption to make. but this is what we adopted in this work. Now, next we do the same. We represent the target domain, the target graphs in these M domains using the feature matrix and also the similarity matrix between subjects. So next, since the data is heterogeneous, so we want to disentangle that heterogeneity. We want to first train a model that is particularly specific to a particular, that captures a particular mode of the data. And this is important because it helps us circumvent the mode collapse in GAN and generative adversarial network, because we focus on a single mode here when we train our network. So here the idea is to basically take the source data to train an encoder, a graph encoder, and then after we get the embeddings in the low dimensional domain of these samples, we cluster them. Next, for the prediction task, we simply train a target specific, a cluster specific set of decoders for each domain. So here we have different domains from T1 to Tm, and the color represents the cluster. So basically for the pink cluster, we train M decoders, and for the yellow clusters, we also train M decoders, but these are all regularized by a single discriminator network. Okay, so the decoders will take in a testing subject. Let's say we take a testing subject, we pass it through the decoder of domain T1 and Tm, etc. So we will get several predicted networks, and then we can simply average them to get the final prediction or the final target network of in that particular domain. So here what we added, what we propose in the paper is an additional topological loss to preserve the topology of the graph. So we look at the topology of the predicted graph and the ground truth one and the target domain, and we try to bridge the gap between them. So for example, we used centrality measures like simple metrics. So here in this example, there are we tested our method on 310 subjects from the Abide data set, and we benchmarked against simple methodologies that were adapted from image-based generation. And you can see that our method, which is called the multi-graph GAN, using different topological measures, it produced the highest PCC, Pearson correlation, between the ground truth and the predicted data, and the lowest mean absolute error between the ground truth and the predicted data. So this was an example of cross-domain prediction. Now, the cross-domain prediction. Now we can move to the cross-resolution prediction. So here, the question that we would like to address is how to predict intermodality non-isomorphic brain graphs from a base graph. So the idea is that we have a simple graph that has a brain network with NR nodes, and we want to learn how to predict a higher resolution network. So generally, the conventional methods, they have, when we do prediction tests from modality 1 to modality 2, from, for example, resting state fMRI to diffusion MRI, we use the same parcellation. Okay, so these are the state-of-the-art methods. They just handle data which are generally isomorphic. So what if the data is not isomorphic? What if we have a change in the resolution of the graph, which means the brain is parcellated, the source brain is parcellated into NR regions, but then we want to predict a higher resolution graph and including NR prime regions at a finer scale. So here, this is what we propose. So the solution, if we look at this, we first propose what we call an intermodality domain aligner. So the proposed architecture called Iman GraphNet is also a graph neural network method. So in the first step, we aim to bridge the gap between the distribution of the target domain and the source domain, because we know that the distribution of these two guys here, they are different because they're derived from completely different neuroimaging modalities. So what we designed is a graph aligner network that takes in the source networks and aligns their distribution to the target to the target domain, which is here in blue. So what we're trying to do is to align the purple distribution with the target distribution. So once we have the target the networks here, they are aligned. So S to T, it means we moved S towards the target domain T, the source towards the target. Then we take these networks, the aligned networks, and we pass them through a non-isomorphic intermodality graph GAN. So this architecture here that we proposed, it takes in the aligned networks and it actually super resolves them, where we can, through these operations with these layers, and we get our predicted super resolved networks in the target domain with higher resolutions. And this whole architecture is regularized by a discriminator there. So we evaluated our data set on the Islem data set, including 150 functional connectomes and 150 morphological brain graphs or connectomes. And we benchmarked against simple methods like and variants also of this Iman graph architecture, because this is actually the state-of-the-art method. This is the first method to super resolve brain graphs across different modalities. So here we use Iman GraphNet without the alignment. So we've got a very high error, as you guys can see. This is the mean absolute error. And then MAE, this is the difference between the input graph and the predicted graph. The MAE between the closeness centrality, which is a topological measure between also the prediction and the ground truth graphs. The MAE between the betweenness centrality, and here we use eigenvector centrality, and this is to see if the prediction, the generation method architecture here is able to preserve the topology of the graph. So we can clearly see that without alignment, we have high error. Then we use statistical alignment. We replace this intermodality domain aligner with a simple statistical alignment method, and we have a drop. We have an improvement in the MAE, which means it decreased. And then once we have Iman, when we tested the Iman GraphNet without the PCCC, we had an additional loss function, which is Pearson correlation. When we removed it, we still see that it is improvement, because here we have, it is improving because here we have the aligner actually. So now without the topological loss, we have also another decrease, but we have the aligner, and then it keeps on decreasing, right, until we get our architecture, Iman GraphNet, including all these topological loss, PCC, and aligner, and achieving the best performance, which was statistically significant using two-tailed pair t-test. So we have included a visual inspection, clinical inspection using random subjects between the ground truth and the predicted graphs. So we picked the top 10 connections, and you can see that they are actually very close. And this is much clearer if you look at the paper and the YouTube presentation. So for the cross-time prediction here, the goal is basically, given a single brain graph acquired at a baseline time point, we aim to predict its evolution over time, the changes in brain and the brain connectivity map. So existing works, actually, they don't generalize to non-Euclidean spaces. So far, existing methods, they focus on image-based prediction or generation over time, not graph-based. So graphs are non-Euclidean structures. And the second limitation is that there are limited works that are so simple, they usually tend to vectorize the methods, the graphs, which leads to losing the topology of the graph in general. So here, what we wanted to propose is basically a method that is template guided, okay, to predict the evolution of a brain graph. So imagine that we have what we call a connectional brain template. So it's like a fingerprint, like of the population that we have at baseline. And using this fingerprint, imagine if we have an average of a population of brain graphs. This average, if we can measure the distance from all other subjects to this average, we can probably be able to predict the evolution trajectory of those individuals, because we do have the evolution trajectory of this average template, of this connectional template, which is purple right here, okay? So the idea here is to use what we call a normalizer network or registration network. And this normalizer network takes in each individual brain subject and aligns it, okay, transforms it into the population template. And through this transformation here, we can pick up, or we can pin down what is individual, what is special about that brain, what makes it different from the population template. So that difference or that deviation, we can use it later on to do our prediction test. So let me explain. So here, once we have our normalizer network that is also regularized by a discriminator, so the CBT is the connectional brain template. I will explain this in the next few slides, how to create a connectional brain template. And so we take these individuals, we align them, and then through this learned embedding here, we can encode the difference between each individual brain graph and the population template. So after that, we compute the residual between each individual brain graph that was basically that passes through this architecture, and the embedding of the purple one of the template, the CBT. And depending on this residual distance, we can then compute the similarity between a testing subject and all these residual training subjects, okay? So this is a residual representation of our training data. And then we can pick or select the k closest neighbors to the testing subject I using this residual representation that is based on this aligner network. And then depending on those distances, we get the closest guys. And once we get the closest training guys, it's simple. We can simply basically pick them up, and then we can get the average all at each time point individually, average the trajectory, average the graphs of those k neighbors, and we can easily get the prediction of the evolution trajectory of the baseline graph. So we tested this on 114 subjects from the OASIS data set, which is a longitudinal data set. And each subject was scanned on 2 or more visits separated by at least 1 year. So we used threefold cross-validation, and we use an independent data set made of 23 randomly selected subjects to create the population network here, the CBT. So we compared with different variations of this methodology without using deep learning. So basically, this is our method, and we have a set of comparison methods, and we report both the mean, the average, mean absolute error, and also the best MAE. And we can see at time point T1, we achieved the best performance, and at time point T2 also we had the best performance, which is the lowest error. So now we go to the last part of this presentation, which is the population brain connectivity mapping. How can we estimate the CBT? So this work was presented at MICAI 2020 and extended into a full media paper, medical image analysis paper. Both the videos and the codes are available. So what is the problem that we will like to solve? So here, given a population of multi-view brain networks or multigraphs, so each subject is represented by a set of brain connectivity maps, okay? Our goal is to learn how to integrate these multi-view graphs or multigraphs into a single graph, a single connectivity matrix, connectivity map that captures the shared traits across subjects, but also across views. So this presents a holistic representation of the brain. So how are we going to solve that? Here we propose the first graph neural network that is able to present a solution to this problem. So first we take these graphs, we use graph convolutional layers and a ReLU function to learn node embeddings for each node or each brain region. Okay, each node here is a brain region. And then after learning these embeddings, B1, B3, etc., for all the nodes in the graph, then we can easily derive the connectivity between them, the pairwise relationship. So here in this example, if the original brain graph data was for example, derived using Pearson correlation, we can use Pearson correlation here between these two vectors to create to estimate the connectivity weight. If we use, for example, absolute difference, we can adopt the same operation. So after that, we will get the CBT, but this CBT is biased towards a single subject that we input to our DGN deep graph normalizer network. Okay, so to give or to estimate a CBT that is for all subjects, what we did, we proposed this new loss. So this new loss function, it computes the distance between the generated CBT for subject S and to all other views. Basically, here we have the view and the subjects. So we try to compute the distance between this CBT and all other views and other subjects in the training set. And here we randomly select a few training subjects to alleviate the computational time. So by optimizing this objective function, we are sure that the CBT is actually centered. So as a further refinement, we take the CBT and that is estimated for each subject individually. Then we perform an element-wise median operation to get the final population CBT. And we tested our DGN on 2 different data sets, small data set, Alzheimer's disease versus late mild cognitive impairment, which had 4 different brain views, and normal control versus autism spectrum disorder subjects with 6 different views. And using the centeredness evaluation, where we see if the CBT is well centered in the data, which means it captures all these shared traces close to everyone at the same time. And we try to maximize its proximity to all these subjects. We noticed that DGN actually, it outperforms state-of-the-art method NetNorm, which is a very basic data analysis method across 40 comparisons. And we also showed that the DGN is able to pin down biomarkers by simply comparing the template, the brain template, the CBT of a healthy population, of a disordered population. You take 2 matrices, you compute their absolute difference, and then you can pick up the regions where with the highest differences. Okay, so for more details, please have a look at the video and of DGN on YouTube, and you can get more details about how this works and about also the exciting results. Now, what's more, we have also this graph neural network review paper in network neuroscience. So all these works are explained and presented in this review paper that is available on GitHub repo. So you can look at all the papers reviewed in this survey that is currently under review. And if you are aware of any works that we missed out, please feel free to drop us an email. Now, let me tell you a bit more in these last few minutes about the BASIRA activities globally. So we are organizing different international workshops and initiatives to advance science and empower research minorities in underdeveloped countries. So there is this WILL initiative, which is about inspirational interviews with leaders and also with students in our field, in the medical field. There is also the RISE network that we started recently as part of the MICAI community to reinforce inclusiveness and diversity and empower researchers in low middle income countries. The FAIR workshop, which is a first initiative at MICAI, so it's a workshop on affordable healthcare and AI for resource diverse global health. The PRIME project, the PRIME workshop is about predictive intelligence in medicine. So we have the 4th edition for this year. And the Progress workshop at ISIP conference for promoting diversity in signal processing. So we need to have the will to rise and make the world fair with PRIME progress. So this is something that we need to consider also as researchers. So if you would like to have more, how do I say it? Like if you would like to get inspired, have a look at the YouTube videos, the WIM-WILL YouTube videos. We have a channel, and these are very, very touching and like moving videos, very inspirational from faculty members, from students, PhD students, postdocs, research scientists, people who have switched from academia to industry and vice versa. So have a look at them. I'm sure you will find a lot of inspiration there. Also the FAIR workshop, you can visit the website and we plan to organize this in future years. So keep this in mind along with the PRIME workshop. which we are organizing this year in conjunction with the MICAI conference. So I would like to thank you all for watching, and if you would like to learn more about our works, you can subscribe to our YouTube channel, and I wish you found this presentation beneficial and inspiring and helpful to your research. Thank you.